Branched chain alpha-ketoacid dehydrogenase complex. The branched chain alpha-ketoacid dehydrogenase complex BCKDC or BCKDH complex is a multi-subunit complex of enzymes that is found in the mitochondrial inner membrane. This enzyme complex catalyzes the oxidative decarboxylation of branched short-chain alpha-ketoacids. BCKDC is a member of the mitochondrial alpha-ketoacid dehydrogenase complex family comprising pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase, key enzymes that function in the Krebs cycle. Coenzymes. This complex requires the following 5 coenzymes: thiamine pyrophosphate lipoic acid coenzyme flavin adenine dinucleotide FAD nicotinamide adenine dinucleotide NAD. Biological function. In animal tissue, BCKDC catalyzes an irreversible step in the catabolism of the branched chain amino acids L-isoleucine, L-valine, and L-leucine, acting on their deaminated derivatives L-alpha-keto beta-methylvalerate, alpha-ketosuvalerate, and alpha-ketoacid respectively, and converting them to alpha-methylbutyl-CoA, isobutyl-CoA, and disobutyl-CoA respectively. In bacteria, this enzyme participates in the synthesis of branched, long-chain fatty acids. In plants, this enzyme is involved in the synthesis of branched, long-chain hydrocarbons. The overall catabolic reaction catalyzed by the BCKDC is shown in Figure 1. Structure. The mechanism of enzymatic catalysis by the BCKDC draws largely upon the elaborate structure of this large enzyme complex. This enzyme complex is composed of 3 catalytic components: alpha-ketoacid dehydrogenase, also referred to as the E1 component, dihydrolipotransacylase E2 component, and dihydrolipamide dehydrogenase E3 component. In humans, 24 copies of E2 arranged in octahedral symmetry form the core of the BCKDC. Non-covalently linked to this polymer of 24 E2 subunits are 12 E1 alpha beta tetramers and 6 E3 homodimers. In addition to the E/E3 binding domain, there are 2 other important structural domains in the E2 subunit: I-lipol-bearing domain in the amino terminal portion of the protein and Roman II inner core domain in the carboxy terminal portion. The inner core domain is linked to the other 2 domains of the E2 subunit by 2 interdomain segments linkers. The inner core domain is necessary to form the oligomeric core of the enzyme complex and catalyzes the acyltransferase reaction shown in the mechanism section below. The lipol domain of E2 is free to swing between the active sites of the E and E3 subunits in the assembled BCKDC by virtue of the conformational flexibility of the aforementioned linker, see Figure 2. Thus, in terms of function as well as structure, the E2 component plays a central role in the overall reaction catalyzed by the BCKDC. The role of each subunit is as follows. E1 subunit. E1 uses thiamine pyrophosphate TPP as a catalytic cofactor. E1 catalyzes both the decarboxylation of the alpha-ketoacid and the subsequent reductive alkylation of the lipol moiety, another catalytic cofactor that is covalently bound to E. E1 subunit. E2 catalyzes the transfer of the acyl group from the lipol moiety to COENZYMES stoichiometric cofactor. E3 subunit. The E3 component is a flavor protein, and it reoxidizes the reduced lipol sulfur residues of E2 using FAD, a catalytic cofactor, as the oxidant. FAD then transfers these protons and electrons to NAD plus a stoichiometric cofactor to complete the reaction cycle. Mechanism. As previously mentioned, BCKDC's primary function in mammals is to catalyze an irreversible step in the catabolism of branched chain amino acids. However, the BCKDC has a relatively broad specificity, also oxidizing 4-methylthioamines-2-oxybutyrate and 2-oxybutyrate at comparable rates and with similar Km values as for its branched chain amino acid substrates. The BCKDC will also oxidize pyruvate, but at such a slow rate the side reaction has very little physiological significance. The reaction mechanism is as follows. Please note that any of several branched chain alpha-ketoacids could have been used as a starting material. For this example, alpha-ketosuvalerate was arbitrarily chosen as the BCKDC substrate. Note steps 1 and 2 occur in the E1 domain. Step 1, alpha-ketosuvalerate combines with TPP and is then decarboxylated. The proper error pushing mechanism is shown in Figure 3. Step 2, the 2-methylpropanol TPP is oxidized to form an acyl group while being simultaneously transferred to the lipol cofactor on EPR. Note that TPP is regenerated. The proper error pushing mechanism is shown in Figure 4. Note the acylated lipol arm now leaves C1 and swings into the E2 active site, where Step 3 occurs. Step 3, acyl group transfer to CoA. The proper error pushing mechanism is shown in Figure 5. Asterisk note the reduced lipol arm now swings into the E3 active site, where Steps 4 and 5 occur. Step 4, oxidation of the lipol moiety by the FAD coenzyme is shown in Figure 6. Step 5, reoxidation of FAD2 to FAD, producing NAD. FAD2 plus NAD plus FAD plus NAD plus H. Disease relevance. A deficiency in any of the enzymes of this complex as well as an inhibition of the complex as a whole leads to a buildup of branched chain amino acids and their harmful derivatives in the body. These accumulations lend a sweet smell to bodily excretions such as airwax and urine, leading to a pathology known as maple syrup urine disease. This enzyme is an autoantigen recognized in primary biliary cirrhosis, a form of acute liver failure. These antibodies appear to recognize oxidized protein that has resulted from inflammatory immune responses. Some of these inflammatory responses are explained by gluten sensitivity. Other mitochondrial autoantigens include pyruvate dehydrogenase and branched-chain oxidoglutarate dehydrogenase, which are antigens recognized by anti-mitochondrial antibodies. Mutations of the BCKDC gene, whose protein product controls the activity of the complex, may result in overactivation of the complex and excessive catabolism of the 3 amino acids. This leads to branched-chain keto acid dehydrogenase kinase deficiency, a rare disease first described in humans in 2012.